These are the recordings for Chapter 5 on large biological molecules, which we often call macromolecules, because macro means large. There are 4 classes of macromolecules. They are carbohydrates, proteins, nucleic acids, and lipids. Carbohydrates, proteins, nucleic acids, and lipids. These molecules that we're going to learn about are, many of them are very large, and they're large chains of smaller molecules. So anytime that we're dealing with really large polymers, we call them macromolecules, just because they are so large. So we have 4 classes of macromolecules. Many of them are polymers, which are long chains, chains of chemicals, and then the individual building blocks, the individual molecules that make up that chain are called building blocks or monomers. So a polymer is a very long molecule made up of building blocks or monomers, and then monomers are the building blocks of large molecules called polymers. So carbohydrates, proteins, and nucleic acids, all 3 of those are polymers. Lipids are large, but we don't call them polymers because they're really not long chains of monomers like the rest of them are. So the carbohydrates, proteins, and nucleic acids are polymers. Lipids are just large. So we'll talk about what makes up a lipid, but the other 3 are long chains of smaller building blocks. Now, we're also going to talk about enzymes, which are actually a special type of protein. But enzymes will speed up chemical reactions, so they are catalysts. They will speed up chemical reactions. So every chemical reaction that forms or breaks down any of these macromolecules is going to have an enzyme involved. There are 2 chemical reactions that we're going to see. One is a synthesis reaction or a dehydration reaction, and then the other one is a breakdown reaction called hydrolysis. So when 2 monomers are joined together to form a larger molecule, that's a dehydration synthesis reaction. And then when polymers are broken down, that is a hydrolysis reaction. And this shows you a dehydration synthesis reaction where you've got a polymer that is being chemically combined with a single monomer. So it had 4 building blocks, and when it's combined with that monomer, it had 3, and now it's got 4 building blocks. So dehydration reactions just add monomers to a chemical. They chemically bond monomers together. And hydrolysis reactions break down the polymers. So the dehydration reaction, it's called dehydration because it joins 2 chemicals together by removing water. So water is formed as a product. And hydrolysis, the hydro is because it uses water, it adds water to the polymer in order to break it apart. So the water splits up into H and OH, right? So the H goes to one of the molecules, and the OH goes to the bonds with the other molecule when they break apart. So dehydration and hydrolysis, just be familiar with those. And we're going to start with carbohydrates. You may know that as a food group. The word carbohydrate tells you the chemical or the elements that compose carbohydrates. Carbo for carbon, hydro for hydrogen, and anytime you have ATE on the end of a chemical name, it means oxygen. So carbohydrates include sugars and what we call complex carbohydrates, which are going to be longer chains of sugar called polysaccharides. So the monomers of carbohydrates are monosaccharides, or simple sugars like glucose and fructose. And the polymers of carbohydrates are polysaccharides like starch, glycogen, cellulose, and we'll go over some more of those. So monosaccharides, mono means one and saccharide means sugar. So monosaccharides, like glucose, are just going to be your simple sugars. There are some that are aldehydes. That means they have the ketone group, the C double bonded to O on the end. And then there are some that are ketoses, or ketone sugars, and they're going to have the C double bonded to O in the middle of the structure, the middle of the molecule. So probably these last three aldoses, ribose, glucose, and galactose. These are all monosaccharides. Ribose is a pentose, which is a 5-carbon sugar. You can count, there's 5 carbons in it. But ribose is the sugar that's found in RNA, which is one of our nucleic acids. The other one is deoxyribose, and that's the sugar found in DNA. So we have pentoses, or 5-carbon sugars, and then we have hexoses, or 6-carbon sugars. Glucose is a 6-carbon sugar, and galactose is a 6-carbon sugar. So these are all monosaccharides. Fructose is a 6-carbon sugar, or a hexose, which is also a ketone because the C double bonded to O is in the middle instead of on the end of the molecule. So glucose is an aldehyde and fructose is a ketone. They're called pentoses when they have 5 carbons. So a pentose is a 5-carbon sugar. A hexose is a 6-carbon sugar. And it's an aldose if the ketone group, the C double bonded to an O, is on the end of the molecule, and it's a ketose when the C double bonded to O is in the middle of the molecule. All right, so monosaccharides are the monomers of carbohydrates. Disaccharides are 2 monosaccharides joined together. That covalent bond is called a glycosidic linkage, the covalent bond that joins 2 simple sugars to form a disaccharide. It's a glycosidic linkage. So when you have 2 glucose molecules chemically joined, here's your glycosidic linkage. You form a disaccharide called maltose. Maltose, or malt sugar, is found in malt beverages, alcoholic beverages like malt liquor. And also the malt that you find in certain candy, like Whoppers, that have the malted milk in the center, and then they're surrounded by chocolate. They're little chocolate balls, but basically they have a malted milk center. Anyway, there's malt sugar in candy, and there's also malt powder that you put in milkshakes. If you order a chocolate shake versus if you order a chocolate malt is a chocolate shake with malt powder added to it. So it kind of tastes like a Whopper shake. But anyway, I'm giving you these examples because I don't know any other good examples. Maltose is a type of sugar, and it's only found in malt, so malt powder, malt candy, malt liquor. Then when you have glucose and fructose chemically joined by glycosidic linkage, you have the disaccharide sucrose, which is the sugar that we find in table sugar. All right, when you have more than 2 monosaccharides joined, you have a polysaccharide. That's many sugars. So one of our polysaccharides is starch. That is a storage polysaccharide in plants. and it's basically just glucose monomers or glucose building blocks formed into chains. The simplest form of starch is called amylose. So starch is a storage polysaccharide in plants. We find a lot of starch in plant roots, especially potatoes and carrots. And then the same type of polysaccharide, but it's found in animals, is called glycogen. Glycogen is basically animal starch. It's a storage polysaccharide that's found in animal cells. In humans and other animals, we find it mainly in liver cells and muscle cells, because what happens is the liver removes glucose from your blood to lower your blood sugar, and it stores that glucose in chains called glycogen. And your muscle cells will do the same thing when you exercise. They will remove glucose from your blood and store it in chains called glycogen. And then when your blood sugar is low, that glycogen can be broken down and the glucose released back into your blood. All right, so here's what starch looks like. Polysaccharide starch. You can see every one of these little green hexagons is a glucose molecule, so it's chains of glucose, and some of those chains are branching. Glycogen is also chains of glucose, and some of those chains are branching. Another polysaccharide is cellulose, also composed of glucose building blocks or monomers. But we cannot digest cellulose. Cellulose is not a carbohydrate that we can digest, but it does help our digestion. It's what we call fiber, insoluble fiber, and we get it when we eat like plant material, like raw lettuce and raw vegetables. Some of this is going into chemistry that down at the bottom, it does talk about how even though humans cannot digest cellulose, for us it's insoluble fiber, which is important in our diet, but we can't actually digest it and get energy from it. But there are some microbes that use enzymes to digest cellulose. Microorganisms that can digest cellulose. And there are some herbivores like cows and termites that have symbiotic relationships with these microbes. They have these microbes living in their gut or their intestines, and that enables them to digest cellulose. But humans are not able to. Another polysaccharide, another structural polysaccharide besides cellulose is chitin. Chitin is found in the exoskeleton of arthropods. Arthropods are going to be like crabs, lobsters, shrimp, and insects, and spiders, scorpions. Their exoskeleton is going to have chitin, going to contain chitin, and also the cell walls in many fungi contains chitin. Okay, so exoskeletons of insects. You see an insect that is molting right now, and then you see exoskeletons of arthropods, which includes insects. And then you see the structural formula for chitin. You also find chitin in the cell walls of fungi. All right, so we have covered carbohydrates. Our next major group of macromolecules are the lipids. And lipids are not true polymers, but they do consist of areas of hydrocarbon chains. And the most important lipids are fats, which can be solid fat or oil, phospholipids, and steroids. So fats are composed from glycerol and fatty acids. And fats are also known as triacylglycerol or triglyceride. Triglyceride and triacylglycerol are the same thing, and that is a glycerol that is joined to 3 fatty acid tails. So a fatty acid is a long hydrocarbon that has on the end a carboxyl group, and that's what makes it an acid. Carboxyl, remember, is C, double bonded to O, and then single bonded to OH. It's COOH. So we have a carboxyl group on the end of this hydrocarbon chain, and that makes it a fatty acid. So when 3 fatty acids are joined to a glycerol, we call it a triacylglycerol or triglyceride. And triglycerides can come in liquid or solid at room temperature. If they are solid, then they consist of saturated fatty acids, which have no double bonds between their carbon atoms. In that hydrocarbon chain, there's no double bonds. But if it's unsaturated, it has at least one, or it can be more double bonds between carbon atoms. So you can see here is an example of a saturated fat, because all of the bonds between the carbon atoms in the hydrocarbon chain are single. But you look here and there's a double bond. Just between 2 of the carbons, there's a double bond. It causes that hydrocarbon to bend. It causes it to be bendy and flexible. And so it is the unsaturated fats that are going to be found in liquid fat or oil, whereas saturated fats are going to be found in, for example, like the butter here, fats that are solid at room temperature. So fats that are made from saturated fatty acids tend to be solid at room temperature and come from animal sources. But when they're unsaturated, they tend to be oils. So they're liquid at room temperature, and they typically come from plants and sometimes fish. Now, you may have heard the term hydrogenated fats or trans fats. Trans fats began as unsaturated fats, but then in a chemical lab, they added hydrogen and made those unsaturated fats into saturated fats. And so it takes unsaturated fats and makes them saturated by adding hydrogen. And that process is called hydrogenation. And trans fats are very, very unhealthy. They're even more unhealthy than a saturated fat is. So margarine, for example, is a trans fat. The fat that's found in regular peanut butter, not natural peanut butter, because natural peanut butter, the kind that you find a layer of oil at the top that you have to stir it up, that is fine. That is unsaturated. But most peanut butter that people buy, the oil is mixed in with the peanut part. And so that is a trans fat, and that's considered very unhealthy.